I spoke about the use of new drugs and new agents in the treatment of childhood acute lymphoblastic leukemia. And it's a very broad topic, so it was a big challenge to try to think about how to summarize, in a sense, the really exciting progress that we've made in the last 5 years in particular. ALL is really the success story of oncology, I think, and pediatric ALL has had dramatic responses in the last 30 years. It went from being really a nearly fatal, universally fatal illness to one where more than 95% of kids can be cured today. The challenge for ALL is that it's severalfold. One is that we have tremendous success, but sometimes at a significant cost for children in terms of long-term side effects. And the first part of that is that when you have an adult with cancer and they survive 20 years past their cancer, if they're 70 when they're diagnosed, that's pretty spectacular. But if they're 5 when a child is diagnosed, just 20 years later is really at the prime of their life. And so as pediatric oncologists, I think we're very focused on the very long-term outcomes for these children. And that's a big challenge because we don't know what the side effects of many of these chemotherapy regimens that we use might be 40 and 50 and 60 years later. So that's the first thing. The second thing is that most of our regimens today have been designed and really kind of settled on because of lots of cooperation across oceans among many institutions and many investigators, with thousands and thousands of patients treated on clinical trials. And we've settled on a combination chemotherapy regimen. We've been able to eliminate radiation for most children with leukemia. And most of this therapy is fairly well tolerated, but again, side effects are significant and not always at a minimal cost for the children and their families. So the second challenge is to consider how can we do clinical trials that actually might allow us to reduce the burden of therapy for children who might be cured with less significant intensity of their treatment. So if we can do that, that's fantastic because we can really tailor the intensity and the aggressiveness of the treatment to the children who need it the most. And we can spare other children a lot of significant side effects and long-term consequences. The third thing is that there's a subset of patients that we still have very poor outcomes from, and those are patients with certain genetic abnormalities in their leukemia that we're able to detect at diagnosis, but we're just learning how to manage them, or we don't have adequate ways of managing them. The subset to that is that they're patients who relapse, even though we think they're going to do fine when they're diagnosed and we think that they have a good outcome. There's some patients that we still can't predict who's going to relapse, and we don't know entirely who those patients are who might benefit from either a completely different treatment approach or more intensive therapy than we already have. So our challenge really is to really risk stratify and to try to give therapies that are appropriate to that exact patient and that exact patient's leukemic abnormalities. Once we're able to do that, then hopefully we can raise the cure rate for everybody and decrease the toxicity burden for the vast majority of children who don't need to have that toxicity. And so my talk really focused on some of the new strategies that are really trying to focus and develop better treatment options for kids. First, for the kids who have relapsed or who have very refractory disease for whom the current treatments are not adequate. There are a couple of categories really to break those down into. The first is immunotherapy, and that would include things like monoclonal antibodies or chimeric antigen receptor T cells. And monoclonal antibodies are kind of a broad category because there are a number of different types of antibody treatments. The most common are antibodies that are linked to a specific abnormality on the surface of a leukemia cell, and sometimes they contain a toxin that's attached to them, so it's like delivering a toxic payload to the leukemic cell. Some of them are actually newer forms of antibodies which engage the human immune system of the patient, him or herself, to better combat the leukemic cell. And those are called bispecific antibodies or trispecific antibodies. And then there's a whole class of treatments called chimeric antigen receptor T cells, which are engineered T cells. They're the patient's own T cells, part of their immune system, that actually can be engineered to be activated against the leukemic antigen on the surface of the leukemia cell. And that's a very novel approach that's really come into its own over the last 3 to 4 to 5 years. The longest patients out who have received CAR T cells are about 3 years out now. And so that's the main category of kind of the immune-based therapies. There are other immune-based therapies that are in development, but they're just a bit younger, and so they're not quite as highly advanced in terms of progression into more widespread use. And then there's a whole series of what we'd call small molecule inhibitors or targeted therapies, which are designed to attack either a protein or an enzyme or something that's unique, a unique mutation in a cancer cell, for instance, that might not be present on the normal form of the normal counterpart of the cell in the human blood system. And those are really a widespread variety. There are hundreds of them literally in development. And we do know that there are certain recurring abnormalities in pediatric leukemias for which some of these targeted therapies can be very effective when introduced appropriately. One of our challenges is to figure out how to introduce those therapies either concurrent with or in some sort of sequence with either immunotherapy or with other more conventional chemotherapy regimens, because we know that conventional chemotherapy works for most kids, and one of the questions is whether or not the new treatments can really synergize with currently available therapies or can be substituted for some of the more toxic currently available therapies and increase the impact of the treatment, decrease the negative impact. That's the million-dollar question. We've actually joked that as we get smarter about sort of subsetting the types of molecular abnormalities in childhood leukemia, there are almost more drugs than there are patients, because we do quite well with the majority of kids with ALL. As we do genomic sequencing, as we learn more about the molecular abnormalities at the very cellular level of an individual patient's leukemia, we're likely going to be able to identify really some personalized treatment options. That gets at the heart of your question, though, which is very complicated, is how do we assess whether or not they actually work? Many of these therapies are fairly expensive, and we want to be able to prove efficacy and safety for them. And the best way we can do that is to do really good clinical trials. Many of those trials have to be international in nature because of the very small numbers of patients who might be eligible for any one treatment. And if that's the case, then you have to stretch your trial boundaries across many international borders to be able to collect enough patients in an appropriately timed way so that you have current data, that you can really assess them in a thoughtful way and hopefully act on the data. Hopefully you can also build into these trials in a variety of ways some companion biology studies so that you are able to figure out if the thing that you think you're inhibiting is actually inhibited, if the target you're actually trying to attack is actually attacked. And then you can really follow those patients hopefully much longer term to be able to determine whether or not that therapy is just a transient benefit or if it actually has a longer term benefit. One of the things that we really, as pediatricians, we have to be cognizant of is that our patients are these growing, developing individuals. They start out, with a 3-kilo baby and grows into an 85-kilo gigantic halting adult. And being able to figure out what happens to normal growth along that way is really critical, because many of the pathways that are abnormal in cancer are developmental pathways. They're responsible for growth, they're responsible for development, and really cancer is uncontrolled growth. And I think any parent that's had a teenager feels like they have uncontrolled growth too, as they're replacing their shoes constantly and the kids are growing out of their clothes in front of their eyes. There are remarkable similarities between a rapidly growing teenager and a cancer cell that's rapidly growing, in that we don't know in cancer cells how to shut them off sometimes. We don't know how to make the cancer cells stop growing under certain circumstances. So if we inhibit the pathways that are making that cell grow, which for a cancer cell is very important, what else are we shutting down? And if we think about skeletal maturation, for instance, or development of the endocrine system, if we think about neurocognitive development, we might be shutting down something that's critical to cancer, but something that's also very critical to normal neurologic development, for instance. And so being able to track those children over the long term, being able to follow their growth carefully, being able to follow their pubertal development, for instance, watching how their cardiac function matures, all of those things are critical to be able to determine whether or not the therapy actually is going to have negative long-term impacts. And unfortunately, those studies take a long time. You can't rush development. The balance, the thing that's trickiest for us and our biggest challenge really is to test the right agents in the right patients at the right time, and to make sure that we do that in a collaborative way across borders so that we can really understand what is happening with the treatment. Is it really what we think it is? And to share that information so that we can advance the field further. I'm always biased that even if a study doesn't turn out to be what you hope the results are, even if you don't cure as many children as you'd like to, hopefully you can learn something from that study that we'll be able to pass on to the next study and the next patients. And I think it's really incumbent upon us as investigators that if a patient is willing to participate in a clinical trial for us, we want to be able to tell that child and that family that their participation really meant something, that it contributed to advancing the field. And so it means that we have to make our trials smart. We have to construct trials that actually contribute to the field and answer questions, even if the ultimate outcome is not that that drug is the right solution. Hopefully that we've learned something along the way that allows us to introduce the next treatment regimen, the next therapy, so that we have advanced things and that we can narrow down the scope of what we're doing so that we can actually have a positive impact on the next children.